All right. Dr. Barry Marshall. Well, this guy is kind of an interesting story. He was a researcher who, back in the late 70s, he was a gastroenterologist who was researching stomach cancer and ulcers, peptic ulcers. And peptic ulcers are associated with stomach cancer. And he discovered that in these people who had peptic ulcers, he was taking cultures of the peptic ulcer and finding that they were florid with this particular bacteria called Helicobacter pylori. It was an acid-resistant bacteria, and he hypothesized that it was the bacteria, in fact, that were causing the ulcer, not the ulcer being an opportunistic opening for the bacteria. Prior to that, people thought stress and like poor eating, like there's the classic scene from like a 70s cop show, the hot dog, like the cop in the cruiser with the hot dog, like throws the hot dog up, ooh, the ulcer, like Starsky, whatever. But that turns out to not actually be the case. So he said this to the medical community, and they thought he was insane. Of course not. This is utterly ridiculous. Everybody knows it's the cheap coney dogs that causes peptic ulcers. So he said, all right. He made a big flask of Helicobacter pylori, just a little cocktail of bacteria, and down the hatch, he drank it up and gave himself a whiz-banger case of peptic ulcers, and then followed it up in kind with a course of antibiotics, and the ulcers resolved in short order. So the medical community stood corrected and was dumbfounded by this. For this act of medical bravery, maybe foolhardiness, he received the Nobel Prize in 2005. So it's a kind of an interesting story, I think, just because it was an example of how what you should absolutely not do actually worked out okay for him. But yeah, it's generally considered to be extremely unwise to experiment on yourself in such a kind of potentially dangerous way. But he was pretty certain, and it turned out that his gamble was correct. Dr. Barry Marshall. All right, so onto the GI tube here. Small intestines. Small intestines are broken up into three parts: the duodenum, the jejunum, and the ileum. The duodenum is by far the shortest, only 10 inches long. However, it's really important in terms of what happens in the intestines. They call it the mixing bowl. This is where we basically change the chemical character of what is at this point called chyme, C-H-Y-M-E. Chyme is the food, fluid, mucus, stomach acid slurry that is what you would call puke if it came out the other end. But if it's heading in the right direction, we call it chyme. So vomitus is the emesis is the medical term for that. So chyme comes in and gets neutralized in the duodenum, and the so-called mixing bowl moves on to the jejunum, where there's a significant amount of chemical digestion and nutrient absorption that continues both through there and into the ileum themselves, which have a combined length of about 20 feet. So what else do I want to say here? There are a couple there are a couple reflexes that we should talk about. We mentioned the gastric reflexes that were part of the myenteric, that engage the myenteric plexus, those short reflexes that cause churning and what have you. But there are two other reflexes. The gastroenteric reflex, which continues the process of peristalsis, or that works on the motility of the chyme through the bolus as it's moving through the intestines. And then when this bolus reaches the end of the small intestine and it's going to pass into the large intestine, there is the gastroileal reflex. The gastroileal reflex is a reflex that opens the ileocecal valve so that the bolus of nascent fecal matter is able to pass into the small intestine. So the vast majority of all the nutrients that get absorbed into the body happens in the small intestine. And then the vast majority of the fluid that's going to be resorbed in the body happens in the large intestine. Right? So because 90% of the absorption of nutrients happens in the small intestine, it's going to be facilitated by a high surface area. All right? So there's this, again, kind of fractal structure of the intestinal lumen. So we have these macroscopic ridges in the intestine. In the bottom left, you can see a picture of the intestine that's been cut open to reveal these things called plica circularis, these sort of fleshy ridges that project into the lumen that greatly increase the surface area of the intestine. And then if you were to look very closely or microscopically at the intestine, that surface, a single one of these plica circularis would be covered in these things called villi. And villi are little finger-like projections that come out from the tissue layer. Villi themselves are not made of a single cell. It's not many villi coming off of a single cell. They are themselves many multicellular structures. The surface of those cells have microvilli on them. So there's another order of increase in surface area even below this picture that I'm showing you here. All right, while we're in the neighborhood, let's catch a little bit of pancreas. So we mentioned the pancreatic. I guess I didn't get a chance to talk about the pancreatic islets in the endocrine chapter. I have a slide I think I chucked in here. We'll talk about it in a minute. But the pancreas is interesting because it has both endocrine and exocrine function. There's several cell types in here. There are endocrine cells and exocrine cells. We'll talk about the endocrine cells in a minute. This is where we get the production of insulin and glucagon. But the exocrine cells are called acinar, A-C-I-N-A-R, acinar cells, the pancreatic acini. And they are responsible for making pancreatic juice. To remind you, maybe you remind me, what is the difference between endocrine and exocrine? You guys remember? Oh, no. Yeah.
Is it one secretes through a duct and one just like secretes through osmosis or something?
Yeah, osmosis is probably not the word I would choose, but I guess there's osmosis.
Can't find the word.
Diffusion into the interstitial tissue that's then picked up by the blood. Which is which? She's got it. Yeah. Which is which? Bloodstream. Yeah, that's right. So these exocrine cells are going to be producing the pancreatic juice and releasing it into this pancreatic duct. There's a long ductwork that runs along the body of the pancreas and dumps into the duodenum through a couple different openings. There's primarily the the duodenal papilla. There's also the accessory pancreatic papilla. Anyways, so this pancreatic juice has pancreatic alpha amylase in it, and amylase is what kind of thing? I talked about amylase last time. What kind of enzyme is amylase? What about starch? It's not actually starch. Starch is amylose. Yeah, it breaks starch down. So starch is mostly glucose 1,4 glucose, alpha 1,4 glucose, and amylase hydrolyzes the alpha 1,4 linkage. So pancreatic alpha amylase. Pancreatic lipase, anyone want to take a guess as to what a lipase is? Breaks fats down, yeah. Ribonuclease and deoxyribonuclease, I think you guys can intuit what those are. They break down the nucleic acids. And then protease and peptidase, zymogens. What did I say a zymogen was last time? An inactive enzyme. It needs to be activated. The one I think I talked about was pepsinogen and going to pepsin. But there's a host of them. We'll talk about that. All right. So first, my first love, carbohydrates. Man, you're so lucky. This is going to be the only slide on carbohydrates I really give you. I would be delighted to teach a whole course on this, but this is what it's come to. So, starch, amylose, comes into the duodenum, and it's this long repeating alpha 1,4 linkage. What you see here is a disaccharide bit taken out of the larger starch thing. Imagine a chain of this that continued in both directions. And then it would have branching with alpha 1,6 branching, but this is a disaccharide component, and in fact we call it maltose. Has anyone ever had maltose before? Where would you get maltose? Does anyone recognize the word? Yeah? Take a guess.
Is there anything to do with malt at all?
Absolutely, it is in fact malt. Yep. Yeah, so if anyone's ever had a malted milkshake, anyone ever had one of those? Or what are those things called? Whoppers? Whoppers. The inside of a Whopper is maltose. And maltose is also in malted barley. That it's one of the sweet components of malted barley that they use to make beer. Anyways, starch comes in. Alpha amylase gets squirted by the pancreas into the duodenal compartment, the duodenal lumen, and it breaks that starch down into the maltose and other oligosaccharides. And there are going to be what are called these brush border enzymes. So I told you there were plica circularis, there were villi on the plica circularis, and the villi themselves are made of cells that have microvilli on them. The microvilli are themselves coated in these, they're decorated in these branched, brushy, fuzzy border enzymes that are going to help break down these sugars. So they're embedded in the surface of the intestinal lumen, in the surface of the microvilli, and they're going to have this contact digestion. So we're not releasing enzymes, they're membrane-bound enzymes that are going to continue to break these sugars down. Maltase, dextranase, glucoamylases, different enzymes that are required to break this starch of whatever sort down into the simple sugars, primarily glucose. There's going to be some other ones, galactose, N-acetylated sugars, mannose. There will be some complicated, complex carbohydrate structures. All right, that's all the sugar you get for today. Well, not exactly, though. This is how we control blood glucose. So we're going to switch over for a moment to the endocrine function of the pancreas. I was going to give this to you in the last series of lectures, but you're getting it today. Because it's important, I think, for you to understand insulin receptor stuff. In fact, I think someone in some group should essentially give this slide to the high school students to give like the larger background of where insulin comes from and what it's doing. But so let's start with lunch or breakfast. We eat some food, amylase, absorb the sugar, blood glucose goes up. The beta cells in the pancreas are going to release insulin. Now, insulin is required, as hopefully those of you working on the insulin receptor have discovered. Insulin receptor is responsible for causing the glucose transporters to be translocated to the surface of the cell so that you can begin to absorb glucose. And this is going to give increased glucose transport into the target cells. And then insulin is also going to have a lot of other effects inside the cell that are part of that diagram I gave you. So glucose is going to get utilized to make ATP. We're going to have a lot more ATP generated for use in the cell. We are going to take excess glucose and store it as glycogen, which is essentially animal starch. Glycogen and amylose are very similar, actually. The only difference between animal starch and vegetable starch is in the branching patterns that they have, but they're all still this alpha-1,4 glucose structure. And this happens in skeletal muscles and the liver. And then it's also going to increase amino acid absorption and protein synthesis. When I talk about energy partitioning later, if I can get to it by the end of the class today, we'll touch on the effects of amino acid absorption and protein synthesis. The concept of energy partitioning relates to percent body fat and the way your body is actually using calories. And then we're also going to increase triglyceride synthesis and form adipose tissue. Now, these last two, the amino acid absorption and protein synthesis and triglyceride synthesis, they're dependent on a lot of other things. First of all, how much exercise do you get? What's your basal metabolic tone? What are the nutritional components you have, like fat to protein to carbohydrate ratios? And not just that, but what kinds of fats? Are you eating essential fats like EPA and DHA, like fish oil fats, or what? And I can talk about that a little bit if we get some time. So, and all of these things are going to lead to the uptake of glucose and the reduction in blood glucose concentration, and we come back to homeostasis. All right, so let's assume now that we're fasting, and glucose level drops. And I don't mean like on the mountaintop with the wind blowing through your hair kind of fasting. I'm just talking about you haven't had a meal in a while. And your blood glucose goes down. Alpha cells are going to produce, in the pancreas, which are also endocrine cells, are going to produce glucagon. And these alpha cells are about 20% of about 1 in 5 endocrine cells in the pancreas are alpha cells, and 4/5 are beta cells. So this is going to lead to the increase in the breakdown of glycogen to glucose in the places that store glycogen, which again are the muscles and the liver. Muscles need glycogen for, contraction, action, and the liver, its job, it mobilizes glycogen so that it can continue to stabilize your blood glucose level, like, for example, while you're sleeping or whatever, and you don't have ready access to calories. It's also going to glucagon also leads to the breakdown, the mobilization of fat stores in your adipose tissue, and the third thing is it leads to what's called gluconeogenesis in the liver, and I'll touch on gluconeogenesis. But all of this is going to make the blood glucose levels go up. This is going to put glucose into the bloodstream and bring us back to homeostasis. Yeah, sir.
This is a little bit off topic, but how much glycogen can we store from glucose before the glucose turns to fat? Or before we
That's a super complicated question, and it's like multi-dimensional answer, and we'll just be patient. I'll get parts of that today, hopefully. All right. Pancreatic enzymes in the duodenum. All right, so let's switch gears to proteins, how we deal with proteins. Again, we've talked about zymogens already. I talked about pepsin and pepsinogen that gets activated by the presence of hydrochloric acid. Well, here's another one. The big one that's the big one in the stomach. The big one in the gut is trypsin, and it's zymogen, trypsinogen. So trypsinogen is a 229 amino acid long peptide or small protein. I guess that's considered a protein. And it has a 6 amino acid regulatory protein. There is an enzyme that is in the gut there called enterokinase that leads to the cleavage, the activation of trypsinogen to trypsin. And once activated trypsin is in the game, it's able to auto-hydrolyze itself. Trypsin causes the conversion of trypsinogen to trypsin. All right, so it begins this positive feedback cascade. But enterokinase is the key that unlocks this. All right. There's a host of other zymogens, chemotrypsinogen, procarboxypeptidase, et cetera, et cetera. Trypsin is able to turn all of these on. Yeah. Oh, did I mention that enterokinase was a brush border enzyme as well? It's membrane bound right in the wall of the intestinal lumen. Anyways, all of these proteases and peptidases are responsible for breaking large proteins down into smaller proteins. All right. So I'm not going to go through the chemistry of that at all, but I'm going to introduce this concept of nitrogen balance. And this relates to proteins because proteins are not the exclusive, but by far the primary source of nitrogen in your body. You can also get nitrogen from nucleic acids and from N-acetylated sugars, but that's minuscule in comparison to the amount of nitrogen you're able to absorb through proteins. Why do we need nitrogen? Well, we need nitrogen to make proteins in our bodies. We also need it, of course, to make we need it to make nucleic acids and the N-acetylated sugars. So the idea of nitrogen balance is if you are in nitrogen parity, that means the nitrogen coming in that in your diet is equal to the nitrogen that's leaving your body through excretion in the urine and through your feces. All right, so we get about 80 grams a day in our food, about like the average person. I don't know, maybe an athlete is going to be eating more than that, but about 10 grams are lost in the feces, whereas most of the urine leaves the body in nitrogenous waste, such as urea, ammonia. I think I showed you the nitrogenous waste in the renal chapter. So the primary store for amino acids in the body is in the body protein. So you have 10 kilograms of your body is dedicated to protein. That's in an average individual. It's a significant amount. All right. So here is positive and negative nitrogen balance. And the top panel is positive nitrogen balance. So this is when more nitrogen's coming in than is leaving the body. And here are some examples of things that lead to that. A bodybuilder who is gaining weight, not a bodybuilder who's at a steady state weight. A bodybuilder who's at steady state weight is probably at nitrogen parity. But if you're putting on muscle mass, you are certainly in a positive nitrogen balance. A woman who is pregnant and growing a fetus is in positive nitrogen balance. A child, as they grow, is in positive nitrogen balance. Speaking of which, we calculated, we mark our children on the bookshelf by my house. We calculated that my 8-year-old son is going to have 4 years until he's the same height as my wife. That was a fun thing yesterday. All right, negative nitrogen balance. That is when less protein is being consumed than is leaving the body. And this happens in, for example, here are some cases that might happen in. On the left is a person with anorexia nervosa. This is an eating disorder that leads to a wasting of the body and a loss of muscle tissue. So people with anorexia nervosa, they're starving themselves to death, and they are literally consuming muscle tissue for energy, which is not what most people do. And then in the middle is a person who is starving not by choice, of course, but in extreme malnutrition, these people are in negative nitrogen balance. And then lastly, any kind of wasting disease, such as there's Stephen Hawking, rest in peace, he just died, who had Lou Gehrig's disease, for example, where the muscle tissue dies and all that nitrogen leaves the body through his urine, actually. So in general, positive nitrogen balance is simply any kind of growth state, and negative nitrogen balance is associated with any kind of wasting away. All right, this slide. God, there's so much more to talk about other than this. Well, these are the chemical events. This is kind of a I cut out all that stuff about lipid transport. You probably saw, some of you that look over my slides, and I figured I would kind of get it a little bit here. Carbohydrates. This is a little bit of a review, putting it all together. Carbohydrates, when you eat them in the oral cavity, they first get broken down by salivary amylase. The starch gets broken down by salivary amylase in the oral cavity. We get di and trisaccharides, which move to the small intestine, where pancreatic alpha amylase comes in with all these other enzymes, lactase, maltase, sucrase, et cetera, et cetera, et cetera. There's a bunch of different hydrolases, hydrolases, glycosidases. The end result of all that is that we get a bunch of monosaccharides that are facilitatively diffused or co-transported into the enterocytes of the intestinal lumen, the cells, the epithelial cells along the inside of the intestine. And from there, they pass directly into the bloodstream and lymphatic system. We're going to come back to lipids in a moment, but I'm going to switch to proteins just because proteins and carbohydrates have a similar motif in that we have large structures that get broken down into smaller structures by enzymatic degradation that happens in the stomach and small intestine to the point where we get the monomers, which in protein is the amino acids, Those are also go through a similar process of facilitated diffusion and co-transport into those enterocytes and then end up in the bloodstream as well. So carbohydrates and peptides, proteins, have very similar methods of absorption. Lipids, on the other hand, is kind of an interesting story. So there's all these lipases. There's lingual lipase in the mouth, the pancreatic lipase. Gets us down to these monoglycerides and fatty acids that form these fat bubbles. And the thing about fat bubbles is they're not water soluble. And so they're not readily exported into the blood directly by the enterocytes. Instead, they form what are called chylomicrons. And a chylomicron is a large glycoprotein and lipid ball that is instead exported into the lymphatic system through a structure called the lacteals. The lacteals are these lymphatic structures that project up into the villi of the intestines, and the chylomicrons pass through the entire lymphatic system and go through something called the thoracic duct, which is the connection between the circulatory system and the lymphatic system in the thorax, and then it heads basically to the liver, where it gets processed. I'm gonna cut that sequence short right there, but it's kind of complicated. All right, because I want to get to the idea of absorptive and post-absorptive states. All right, the absorptive state is the period during which you've eaten. All right, you've eaten some food, your body is processing that food, you're absorbing the nutrients. So here we go on the left at 8 a.m. You've had breakfast before going to your first class, and your amino acid, your lipid, and your glucose levels in your blood are at their peak. And with time, over the next 4 hours, they're going to drop down until around noon, where you will briefly go through a post-absorptive state, which is going to trigger appetite. And you're going to have another meal. You'll have lunch, and that will spike once again across the 3 nutrients. Again, that will decline over the next 4 hours or so until you have a short period of a post-absorptive state, where you'll have a third meal. And that'll decline until it's time for bed. So during the absorptive state, on the cellular level, you are going through more anabolic processes than catabolic processes. Anabolic, anabolism, is the process of building things. Cells build things during anabolism. And catabolism is breaking larger structures down into simpler structures. That's all. So during the absorptive state, anabolism dominates, and the fuel for this, what we use to drive anabolism, is glucose, which will get converted into ATP. On the other hand, the post-absorptive state is after the complete absorption of nutrients in the GI tract, and this is when catabolism exceeds anabolism. So there's no more food in the GI tract. Our circulating levels of macronutrients has bottomed out, and we're going to be going through catabolic processes, breaking things down more than anabolism. When we do this, the fuel that we use to produce ATP is glycogen, which is animal starch, carbohydrate, glucose repeats that are stored in the skeletal muscle and the liver, and fat. So we burn fat during the post-absorptive state. Here's a little bit of an overview. And all of this is an introduction into cellular respiration, which is what I'm going to talk about, I guess, for the rest of the class here. So these macronutrients, I'm calling them organic molecules here on the left. They represent the three categories: proteins, lipids, and carbohydrates. Proteins, fats, and carbs. These amino acids, lipids, and simple sugars are absorbed into cells in the body, and they enter what's called the nutrient pool. And from the nutrient pool, two things can happen. You can either go through anabolic processes and build larger structures. That means like the maintenance and repair of the cell, or growth, or secretion of some kind of thing, an epithelial cell, or maybe stored reserves of fat or glycogen, something like that. Those are all anabolic processes. Or elements of the nutrient pool can go through a catabolic process and get shunted into central metabolism, glycolysis, and go straight to the mitochondria, where we get the production of ATP. ATP, about 40% of the energy that's released in the mitochondria is captured as chemical energy in ATP, which is going to drive all kinds of things. It's going to drive various anabolic processes and any other kinds of functions that require energy in the cell. About 60% of that energy, chemical energy, is just lost as heat. It's just lost as heat. Questions here? All right, so I'm going to fill in a few of the details. Let's just zoom in on the mitochondria just a little bit more. I'm going to continue to add details to this. So here's the nutrient pool inside the cell: the fatty acids, glucose, and amino acids. They're anabolic products, so you can make triglycerides from the fatty acids. Glycogen is the way that we store glucose, and proteins obviously are made of amino acids. All three of these nutrients, small nutrients, can make small carbon chains like acetyl-CoA and pyruvate, which are then able to be imported into the mitochondria and enter the tricarboxylic acid cycle. And then these coenzymes, these reducing equivalents that the TCA cycle makes, are shunted into the electron transport system, which is where we make ATP. The output of this is carbon dioxide. All right. So we're taking these small carbon chains and fully oxidizing them, and then releasing carbon dioxide. All this energy that's being captured by the mitochondria is stored in the formation of this phosphodiester bond that you see here at the terminal phosphate in adenosine triphosphate. It's that bond that is with the arrow there that's going to be cleaved to release the energy inherent in ATP. Hopefully that's a review for everybody. All right. So here's the overview of all of this, all the catabolic and anabolic processes. We have all the anabolic processes in blue, that's the building up of things, and the catabolic in red. So again, there's this nutrient pool level: the fats, the proteins, and the carbohydrates, or the amino acids and the carbohydrates. And then on this top level are these larger structures: the triglycerides, the glycogen, and the proteins. Right down the middle is what we call central metabolism. The breakdown of glucose to pyruvic acid is what we call glycolysis. The buildup of glucose from pyruvate. We can make glucose from small carbon chunks that maybe we can get from fatty acids or amino acids. This is called gluconeogenesis. And then going above that, from glucose to glycogen, breaking glycogen down is called glycogenolysis, and making glycogen is glycogenesis. from glucose. All right. So now we're going to put it together. That picture I just showed you is not the same at all points in time. Certain aspects of these pathways are emphasized during the absorptive versus the post-absorptive state. So here's the absorptive state right after you've eaten. And as I said, we're emphasizing all of the anabolic pathways in the cells. The one constant catabolic pathway is glycolysis because we're always going to be trying to produce some ATP that's required. The nutrient pools are feeding either into central metabolism to give us the output of ATP, or they're going to build larger structures, the protein, the glycogen, or the triglycerides. You can see there's a host of different factors that influence this. The one I would highlight is the impact of insulin on protein building and glycogen storage and the formation of triglycerides. Also, the conversion of acetyl-CoA to fatty acids. So you see here that insulin has effects all over the cell. That's detailed much more in that slide that you guys will be talking about at the end of the year. So the post-absorptive state is just the reverse of this. All of the catabolic pathways are getting stimulated, and glucagon, you'll note I'll highlight glucagon, which is all over this slide, is involved in activating several of these pathways. In terms of central metabolism, the glycolysis is still there, but it's not as important as gluconeogenesis, the making of new glucose from pyruvate upwards. All right, so is there one take home that I want to give you guys here? I don't think so. I think that's it. That was pretty much all I was going to talk about.